We will talk about neural tube development and how you form vertebrae, how you form your bones around your spinal cord. But almost everything we're talking about from now on until the end has to do with vertebrate development. Okay, and you have to understand the fundamentals of the dorsal organizer with respect to vertebrate development, because that's where it's all going. Right, so while, the idea of the dorsal organizer, I'm not going to test you directly on the dorsal organizer on the final exam, you have to understand why the dorsal organizer is so important, because it's crucial to making the spinal cord and the vertebrae, which is what we have. Okay, so the dorsal organizer is that little autonomous region that's sort of found where gastrulation begins. It's found opposite the spur-entry point, and we'll talk about that today. And that's this region, gastrulation begins there, and you have cells moving from what will be posterior to anterior, and they become this dorsal structure called the notochord, which we'll talk about today. Okay, so this dorsal organizer region, or Spemann organizer, or Henson's node in chick, or just the node in us, it's this dorsal organizer that will make this thing called the notochord. So there's 4 functions of the dorsal organizer. One of them is it will become dorsal mesoderm. It will become this thing called the notochord, which is going to be found on the dorsal side. So if you do a cross section, this is an embryo that's finished neurulation. That notochord is responsible for making neural. So you have dorsal neural because you had a dorsal notochord. And we'll see how that works the next couple of lectures. Okay, so the dorsal organizer is going to form dorsal mesoderm. It's autonomous. If you transplant it to another embryo, it'll form another axis. It'll form another dorsal axis. Okay, so it self-differentiates into dorsal mesoderm. It initiates the movements of gastrulation, right? If you put it in another embryo, you're going to get another dorsal lip, and you're going to form another axis. It can dorsalize surrounding mesoderm, and it can dorsalize ectoderm into neural ectoderm. So the idea of this thing dorsalizing, what the heck does dorsalizing mean? It means it's more dorsal, it's more towards the dorsal side. So if you have an embryo here, this embryo has an anterior at one end where the mouth is, a posterior at the other end where the anus is. In this diagram, the top side is the dorsal side, that's where the neural tube is, and the other side is the ventral side. So dorsalize means it's going to put it more into the dorsal axis. So the closer you are to the notochord, the more dorsal you are, right? So the ectoderm that's going to be close to the notochord is going to form neural. So the ectoderm that was close to the notochord is going to become neural ectoderm, which is going to form the spinal cord. And we'll talk about that in a lecture or two, right? The closer you are to this dorsal notochord, the more dorsal you are, because this notochord is secreting molecules that make things dorsal. Right? The further away you are from the notochord on the ventral side, you're not getting these molecules. It's literally secreting ligands. We'll talk about them today: noggin, cordon, follistatin. It's secreting a bunch of things. The closer you are, the more dorsal you're going to be. The further you are, the more ventral you're going to be. Right? If you don't get those signals, you're going to be ventral and you're going to form blood. If you're closer, you're going to form things like the urogenital system, your kidneys. If you're closer, you're going to form muscle. And if you're right at the end, you're going to form vertebrae, your bones. And we'll talk about that. But the idea of dorsalization means you are close to dorsal, so you have more dorsal character. Right? The types of tissues that you find at dorsal. Right? And again, there's all these molecules that we're going to be talking about that do all this. Is that good? So, let's go back to this diagram. So this is a little diagram that's showing you sort of the outside of the embryo. This is showing a cross section, and then this is showing you what will form the mesoderm. Right? So this is sort of this layer of tissue that during gastrulation is going to form the mesoderm. It's showing you gastrulation and the dorsal lip on the left side. I like the dorsal lip on the left side, so I always flip this thing. Right, so now the dorsal lip is on the right side here, which is sort of how I normally draw my diagrams. So of course on exams, it can be flipped in all kinds of ways. Okay, so the dorsal lip here is on the one side. That means the spur-entry point is on the other side. So there's something special about this dorsal organizer. It's going to start gastrulation. If you transplant it, it's going to self-differentiate. It will form the dorsal mesoderm. Right? anything near it is gonna become more dorsal, it'll dorsalize surrounding the mesoderm, or it's gonna make the ectoderm near it more neural. Okay, so it's all about trying to understand what's happening with this ring of mesoderm. Okay, this is sort of useful, all of the colors are somewhat confusing, but if you kind of look at this diagram, this is showing you the outside of the embryo. So in this sort of light blue, dark blue, yellowish color, this top part, you can see here it says non-involution widening zone. All this is ectoderm, right? What happens during gastrulation is because of the pivot, all this ectoderm ends up closing at the blastopore, which is posterior. So what you end up with is just ectoderm on the outside, which makes sense. So there's ectoderm on the outside, the mesoderm is going to be the next layer, and then the endoderm is going to be inside that. And this is showing you all the layers of that. The point of this is trying to show you that this ectoderm is not all equal, because the ectoderm that's associated with the dorsal side is different, because it's going to form the spinal cord, it's going to form neural tissue. So the ectoderm associated with the notochord is going to become neural, and we'll see how that works. Okay, so let's kind of just focus on this mesoderm layer, right? So if you take that mesoderm layer and kind of look at that on its own, specifically these two little regions here, what are they trying to show you here? They're trying to show you that in this purple region, these purple cells, that is the dorsal organizer. Those dorsal cells are different. If you remove them, they will form dorsal mesoderm no matter what. They're autonomous. During gastrulation, these purple cells flip themselves inside out through involution. This whole band of mesoderm sort of flips itself inside out. But most importantly, these purple cells, this dorsal organizer, they're going to form the dorsal side of the embryo. So the cells that were the dorsal organizer are now this strip on the dorsal side of the embryo. Right? It's showing you this orientation, but if you just flip this 90 degrees, right, this is basically the same as the little embryos we were looking at just now. Right? You have posterior on one side, anterior on the other side. This dorsal organizer region is undergoing involution and moving into the embryo. That's the same as this little red area here. So the mesoderm that's from the dorsal organizer is moving along the dorsal side. So this purple is that mesoderm. Right? There is mesoderm moving along on the ventral side, this mesoderm here, but it's not dorsal mesoderm, that's ventral mesoderm. Eventually, once you get towards the anterior, the dorsal organizer cells are going to be forming this dorsal mesoderm. It's going to form the notochord. Okay, so this is looking at just the mesoderm, and this is looking at the whole embryo. Right? If you take a cross section through this mesoderm here, you're basically going to be seeing this circle of mesoderm. There's dorsal mesoderm on one side, ventral mesoderm on the other side. The reason that this dorsal mesoderm is dorsal is because the notochord is here. So these arrows are talking about a whole bunch of different things, but most importantly, you can imagine these purple cells are secreting molecules. The closer you are to them, the more dorsal you are. So the mesoderm that's right next to it is going to be more dorsal, and as you get further away, it's more ventral, because you're further away. Right? So the dorsal organizer has four properties. It initiates the movements of gastrulation. It becomes the dorsal mesoderm. It becomes this thing called the notochord. It dorsalizes the mesoderm near it, and it also dorsalizes the ectoderm above it, so that the ectoderm above it, dorsal to it, is going to form the neural tube, neural tissue. So eventually you're going to form the neural tube on top of it, and then eventually you'll form the vertebrae around the whole thing. And that'll be your spinal cord. Your vertebral column, I should say, with your spinal cord in it. So far so good? Ish? So the big question is, what the heck is, why do you have this dorsal organizer? What is special about the dorsal organizer? We talked about that it occurs opposite the sperm entry point. Alright? So what's happening opposite the sperm entry point to make this dorsal organizer? Okay, so lots of animals do it in different ways. This is how frog does it. Right? frog does it with something called cortical rotation. I mentioned this before. The outside of the fertilized embryo rotates with regards to the inside. So this is a fertilized zygote. Okay? So it has a cortex, has an outside bit of cytoplasm that's different than the inside cytoplasm. Right? So the outside cytoplasm has the pigment cells in it, and it's a little bit different. Okay, so there's a layer on the outside of the zygote that's a little bit different than the layer on the inside of the zygote. The pigmented cells are on the outside. After fertilization, I think I mentioned already to you that there's a rotation. The outside, the cortex, rotates with respect to the inside so that the pigment cells actually rotate, and then you swirl material. You saw that one picture of that sort of false color thing. There's a swirling that occurs, and because that swirling occurs in Xenopus, at least in frogs, you end up with this gray crescent. So there's a little region where you've mixed pigment cells. So you used to have, so there's normally pigmented cells on the animal half, unpigmented yolk cells on the vegetal half. But after cortical rotation, you still have pigmented cells on one side, you have yolk cells on the other side, but you have this region now, because of the swirling, you've mixed pigment, and now you have this thing called a gray crescent. That just shows you that you've had movement. And we already know that the sperm entry point goes opposite the gray crescent, and then the first cleavage plane is going to go right through that gray crescent. That gray crescent is where you're going to form the dorsal organizer. Everyone got that so far? So what is it about sperm entry point that's causing this? Short answer is the sperm entry point is not only delivering the nucleus, so when the sperm fuses with the egg, it's delivering the nucleus, but it also delivers one of its centrioles. The sperm centriole is going to enter into the egg, right? With that sperm centriole, it's going to repolymerize microtubules that are inside the egg. Okay, so a centriole, if you remember, is a microtubule organizing center. So before fertilization, or just right after fertilization, if you look at the microtubules in the cortex, the microtubules are disorganized. After you get the delivery of the centriole, you can see that the microtubules are repolymerized. Okay, so there's a change in the polymerization of the microtubules according to that sperm entry point and that centriole. Okay, they're not really showing you that well here, so I'm going to try to draw that a little bit better over here. All right, so you have an egg. The outside is a little bit different. It's called the cortex. Okay, inside this cortex and sort of outside it, you have loosely organized microtubules. There's microtubules that are loosely organized before fertilization. When fertilization happens, sperm comes in, it's going to deliver the nucleus, but it's also going to deliver a centriole. That centriole is a microtubule organizing center, and what it's going to do is it's going to reorganize these microtubules, and the simplest way you can think of it is it's going to reorganize them downward with gravity. So if gravity is this way, it's going to polymerize it with gravity, and you're going to polymerize all of these microtubules going down with gravity and up the other side. So all the microtubules, not all of them, but most of them, are going to be polymerized. If you remember, and I'm sure you do from second year cell biology, this is going to be the plus end of the microtubules going away from the centriole. So the plus end is going to go down with gravity and then up the other side. Make sense? So this is sort of near the cortex, near the outside of the embryo. They're not quite showing you that there, but they're trying to show you that there's a difference. So this is sort of before fertilization or just after fertilization, a little bit later, there's this orientation. What you're really doing is you're polymerizing them close to the cortex. There are going to be other microtubules, so these aren't all the microtubules. Eventually you need mitosis to occur, you need karyokinesis to occur, so there's going to be other microtubules. But these microtubules are going to play a role in rotating the cortex. They're going to rotate things 30 degrees. So the outside of the egg is gonna rotate with respect to the inside of the egg. All right? So, what do you got? As fertilization is occurring, you have pigment cells on the top, so you have the animal half, and you have the vegetal half that doesn't have any pigmented cells. What you also have is lots of maternal determinants. We've said this lots of times, there's all kinds of maternal determinants in here. One of the things that's a maternal determinant is disheveled. The protein disheveled is on the vegetal side of the egg. Okay, and now it's on the vegetal side of the zygote once fertilization occurs. Okay, so down here, you have disheveled. It's just vegetally located on the vegetal side. That disheveled is inactive, so it's inactive disheveled. What then is gonna happen when fertilization occurs, you're gonna donate a centriole. That centriole is gonna polymerize microtubules. Those microtubules can then be used by motor protein. What motor protein? Kinesin. Okay, so kinesin, if you remember, is a plus N directed motor. Kinesin is going to move towards the plus end of the microtubules, and kinesin is going to rotate the whole cortex. It's rotating the outside of the egg with respect to the inside of the egg. Okay, so that whole cortex, the cortical elements, the whole outside of the egg is going to rotate with respect to the inside of the egg about 30 degrees because kinesin is moving things. That make sense? That movement is going to cause the swirling of the cytoplasm, and it's going to cause the movement of some but not all maternal determinants. It will cause the movement of disheveled. Disheveled is going to be moved away from this vegetal side up towards opposite the sperm entry point. Okay? The amount that disheveled moves is actually more than the cortex, and that's because disheveled is also actively being transported. So not only is kinesin moving the cortex, but kinesin is also actively moving disheveled. So the amount kinesin moves is actually more than 30 degrees, it's probably more like 45 degrees, but regardless. This makes sense? Okay, so now you have disheveled, and it's localized opposite the sperm entry point. Kind of cool. What does disheveled do, if you remember way back when? Disheveled is part of the Wnt signaling pathway. Normally disheveled is activated by Wnt, and they have Wnt in here, but we're not quite sure exactly what role Wnt is playing because this is one egg. Normally, Wnt signaling is juxtacrine signaling. You need something to release it, you need a receptor. It doesn't look like it's happening here, so we're not exactly sure, but regardless, disheveled is present, and disheveled is going to do its function. And what does disheveled normally do? Disheveled normally inactivates GSK3. So, backing up a little bit, the whole egg, the whole zygote has GSK3 everywhere. So there's GSK3 present everywhere as a maternal determinant. So there's lots of maternal determinants, some of them are localized, disheveled, some of them are not localized, it's just GSK3. If you remember what GSK3 does, GSK3 targets beta-catenin for destruction. So if you have GSK3, it will degrade beta-catenin and you will have no beta-catenin. So because of disheveled now, on one side of the embryo, you have disheveled is on one side of the embryo. There is no disheveled on the other side of the embryo. So opposite the sperm entry point, you have disheveled. Disheveled inhibits GSK3. GSK3 now cannot degrade beta-catenin. On the other side of the embryo, GSK3 is active. So GSK3 can degrade beta-catenin. Taking a step backwards, I should have also said that beta-catenin is maternally distributed largely evenly through this vaginal half. So there's beta-catenin everywhere in the vaginal half. We've seen that before. Beta-catenin is in lots of vaginal cells. It's in there with the tunicate. If you remember, there's beta-catenin in those vaginal cells. So beta-catenin is in the vaginal cells, but now it's going to be degraded on one side. Beta-catenin is only going to be present opposite the sperm entry point. So all of this occurs before the first cell cleavage. All of this occurs in the first 20 minutes. Right, so you have microtubule polymerization, you have catenin, kinesin moving things, you have disheveled on one side, disheveled is activated, disheveled inhibits GSK3. GSK3 cannot degrade beta-catenin on one side, GSK3 can degrade beta-catenin on the other side. This is all before the first cell cleavage. Then the cells start to cleave, and they cleave very quickly. When you look later on, by the time you get to the blastula, you have a blastocele cavity, right? Now you have cells on one side of the embryo, opposite the sperm entry point, they have beta-catenin. The cells on the other side of the embryo, where they're not opposite the sperm entry point, don't have beta-catenin. So this is where gastrulation is going to begin with mid-vellular transition. The cells of the dorsal organizer have beta-catenin, so when they begin to transcribe, they have these maternal determinants, these transcription factors that can turn on genes that cause cell movement and make these things notochord. It's more complicated than that, and we'll see that in a second on the next slide. But does everyone get the basics of how you get beta-catenin opposite the sperm entry point? That good? All right, how do you know this is sort of what's going on? Well, you can start playing with different things. For example, if GSK3 plays a role in getting rid of beta-catenin, what happens if you inhibit GSK3 on the other side? Normally GSK3 is active over here. If you inhibit GSK3 or you inject a dominant inactive form, right? If you kind of mess with GSK3, you're going to allow beta-catenin to be present in other places, and wherever beta-catenin is present, it's going to form another axis. Right? Because beta-catenin is present in lots of other places. There's a couple of other things that's important in terms of trying to understand what's happening, and that is treating embryos with ultraviolet light. So after fertilization, you have these microtubules being polymerized, right? And this happens within the first 20 minutes. If after fertilization, you take an ultraviolet light and shine it on this vaginal side, so you have this ultraviolet light shining from the bottom of this embryo. Ultraviolet light ultraviolet light prevents microtubule polymerization. So if you use ultraviolet light, you will not get microtubule polymerization. If you don't get microtubule polymerization, you will not get cortical rotation. If you don't get cortical rotation, disheveled is going to be left where it is, and it's inactive. If you don't have disheveled, GSK3 is active everywhere. So if you treat with UV light, GSK3 will be active everywhere. There will be no beta-catenin everywhere. That means you will get no dorsal. So treating with UV light, you end up with something called a ventralized embryo, because there is no beta-catenin everywhere. Why? Because there was no cortical rotation. That means the disheveled didn't move. That means it did not inhibit GSK3 anywhere. So GSK3 was everywhere, which means beta-catenin is nowhere. That means no dorsal. So the whole thing is centralized. And then you can do experiments to try to rescue it. What do you need to rescue? If you inject beta-catenin back in, wherever you inject it, that will become the dorsal organizer. The other treatment that's opposite is treating with lithium chloride. Lithium chloride inhibits GSK3. So if you treat an embryo that's just been fertilized with lithium chloride, no GSK3 will be active. So there will be no active GSK3 anywhere. That means beta-catenin will be everywhere. That means beta-catenin will try to make everything dorsal, and that's called a dorsal embryo, dorsalized embryo. Right? It's going to try to make everything a notochord. Okay? This is very powerful. This is what we do. That's why lithium is used in lots of drugs, and on every drug with lithium in it, there's a huge warning for pregnant women, do not go near lithium. This is why, because during development, you need, lithium can mess up dorsal organizer, can mess up dorsal structures. So lithium, which is not toxic per se, but if you're pregnant, it's gonna affect, your embryo, it's gonna dorsalize your embryo, which is bad. Okay, so you have cortical rotation that's involved in making dorsal because that's where beta-catenin is. But you can treat embryos with ultraviolet light to ventralize them because you don't get cortical rotation. Or you can treat with lithium chloride, which inhibits GSK3, that means beta-catenin is everywhere, so everything is dorsal. Cool? All right, so more specifics. Let's get into some signaling. Okay, so you have a lot of maternal determinants. During cortical rotation, maternal determinants move or maybe not. So some maternal determinants are going to move, some maternal determinants are not. Okay, so that's an important part to remember. Okay, so by the time you get to the blastula stage, you have different patterns of maternal determinants, and this is what it's trying to show you here. Okay, so for example, at the blastula stage, just before mid-blastular transition, I already told you, initially you had GSK3 present everywhere. Right? So GSK3 is not affected by cortical rotation, but GSK3 is going to be affected by disheveled levels. Right? But if you look at the embryo at the blastula stage, so this is just before mid-blastular transition, just before gastrulation is going to begin. On one side of the embryo, you have beta-catenin. That beta-catenin is opposite the sperm entry point. Why? Because cortical rotation occurred and disheveled protected beta-catenin over here. Okay, so again, this is not a one-cell embryo. This is a 4,000 cell embryo just before cortical rotation. Okay, these maternal determinants were like this before cells started to divide. Some maternal determinants moved, such as disheveled moved. You can see there's this thing called XNR, which is Xanthus nodal related. This is a protein called nodal. It has a slightly altered distribution. Other maternal determinants, such as this VEG-T, VEG-1, did not move because of cortical rotation. So you have different maternal determinants, some of them that moved with cortical rotation, some of them that moved a lot, some of them that moved a little. What you end up with, though, is this overlapping picture of things. So if you look at opposite the sperm entry point, what is opposite the sperm entry point? Opposite the sperm entry point, you have high levels of beta-catenin, high levels of Xanthus nodal related, and VEG-T, VEG-1. Okay? And it's this overlap of these different things that give you the dorsal organizer. It's not just beta-catenin by itself, it's beta-catenin and other signals that are present there. Okay, so if you look at things, and this is sort of important, and hopefully it'll become clearer once we go through the next bunch of slides. Okay, so some cells are going to have beta-catenin, but some cells will have beta-catenin and Xanthus nodal related, and they will be under the influence of VEG-T and VEG-1. Those are going to be the most dorsal. They are going to give rise to the organizer, the dorsal organizer. So those cells, if you transplant those, they will start gastrulation and become dorsal mesoderm wherever you put them. Other cells present are going to have VEG-T, some of them will have nodal related. They're going to give rise to different types of mesoderm. So there's going to be other signals going on. If you remember, there's this band of mesoderm. So you're going to form different types of mesoderm, but only the dorsal organizer is going to have the combination of beta-catenin, nodal related, and VEG-T. Okay, the other cells here are going to give rise to different types of mesoderm, like ventral mesoderm. That's so far so good? Okay, so how do you know what you have? You're looking at specific genes. So for example, one of the master genes for the dorsal organizer is a protein called goosecoid, which we'll talk about on the next slide or slide after that. Okay, so goosecoid is present in the dorsal organizer. It's one of the first things that kind of defines the dorsal organizer. Okay, Then you will see these other genes, C1 and TGF-beta family molecules that we'll talk about later. Right, so when you look at sort of this little diagram we talked about before, you have this band of cells that are going to become the mesoderm. These cells are going to have different fates. Some of them will form ventral mesoderm because they're sort of on the ventral side. The other ones are going to be dorsal mesoderm because they have beta-catenin and xanthus nodule related and VEG-T and VEG1. Those are the ones that are going to be the dorsal organizer, right? So the ones that are close to dorsal, they're going to try to make everything else more dorsal. They're going to be signaling to make things dorsal. If you're far away from dorsal, you're going to be ventral. All right, signaling, what the heck is actually going on here? So there's a lot of players here, a lot of things that are present, and a lot of them are in the TGF-beta superfamily. So if you remember this TGF-beta superfamily, you have these TGF-beta molecules as well as BMPs, and the differences whether they signal through SMAD1.5 or SMAD2.3. So they play important roles. In this family are also VEG1 and nodal, so these are also TGF-beta molecules that are going to signal through SMAD. Okay, activin is also present here. If you remember before, I mentioned when people were looking at how mesoderm induction occurs, they would take the animal cap cells. You can take these ectoderm cells, and if you take these ectoderm cells and treat them with different levels of activin, you can form different types of mesoderm. You can form the notochord, or you can form muscle, or you can form blood. And that's because activin is present. Activin is present down here, and it does signal, but it's not as important as some of these other molecules. So when people started to biochemically isolate proteins from the endoderm, for whatever reason, they isolated lots of activin, and activin does do the job, but activin actually does not play an important role during gastrulation. It's at very low levels. The important role is played by nodal and VEG1. Okay, so what's happening here? Okay, so you have cells, and now you're starting the mid-blastocyst transition, which is zygotic transcription is going to occur. So you've gone, in the case of Xenopus, through 12 cell cycles. You have 4,000 cells, whatever it is. Now zygotic transcription is going to be turned on. Now you're going to be using the nucleus and whatever maternal determinants are there. So different cells are going to have different signaling molecules. For example, these cells are going to have beta-catenin. Other cells are not going to have beta-catenin. Right? There's another protein that's present everywhere called TCF3. TCF3 is involved in beta-catenin signaling. Without beta-catenin, it represses transcription. Okay, so genes that are supposed to be dorsal genes are repressed in the absence of beta-catenin. So most of the embryo doesn't have beta-catenin. TCF3 is going to repress dorsal genes. Where there is beta-catenin, when zygotic transcription begins, beta-catenin will bind with TCF3, and now you're going to activate dorsal genes. One of the first dorsal genes activated is a gene, two very similar genes, one called C1 and another one called twin. From the name, like Siamese twins and twin, these proteins are involved in early axis formation. Just like beta-catenin, if you take beta-catenin and inject it into another embryo, you can form another dorsal axis. You can take the same thing with C1 twin. They will form the dorsal axis because they are the first step down in beta-catenin signaling. So beta-catenin is going to turn on, it's going to turn on C1 twin. C1 twin are then going to be working with the other superfamily molecules, okay, mainly nodal and VEG1. So where are you going to find nodal and VEG1? Well, in that area of the dorsal organizer, right? Backing up one, oops. Right? It's where you have VEG1 and nodal and beta-catenin. That's where you're going to have the dorsal organizer. Okay? So in terms of transcription, VEG1 is going to turn on C1 twin, but then they need, beta-catenin is going to turn on C1 twin, but then they need nodal and VEG1 to turn on the subsequent genes. And it's only at this step that you make goosecoid. Once you have goosecoid, you've made the dorsal organizer. Right, so it's essential for turning on other genes. Goosecoid is a transcription factor. All right, so you have an overlap of signaling molecules present in the dorsal organizer, beta-catenin being in the nucleus. It's going to turn on C1 twin, and then in those same cells, you're going to have nodal and VEG1, which are TGF-beta superfamily molecules. They're going to be also responsible for then bonding into and turning on downstream genes, with goosecoid being the first one. Once you get goosecoid, everything then continues. You're committed. You're autonomously dorsal organizer once you have goosecoid. You then start turning on other genes such as noggin and cordon, which we'll talk about now. Everyone get that? So only the cells of the dorsal organizer are going to have goosecoid, basically. Other cells may be mesoderm, but they're not going to have goosecoid. All right, so goosecoid is defining, it's a transcription factor that defines it. When you look at goosecoid, it makes sense to be the master dorsal organizer molecule, right? It autonomously determines dorsal fate of the cells expressing it. So if you have goosecoid, you will be dorsal mesoderm. You will become the notochord. It can make other things more dorsal. If you're close to a goosecoid cell, you will become more dorsal. So when you look at an embryo, so when you look at where goosecoid is going to be found in the dorsal lymph. That makes sense. So goosecoid mRNA is found in the organizer, and then the protein is found in the organizer. That makes sense. Beta-catenin is turning it on. Goosecoid is present where it is. If you inject goosecoid into another embryo, wherever you inject it, you can form another axis, a secondary axis. That's just like transplanting the dorsal lip, because that's what you're kind of doing. If you UV treat an embryo, if you UV treat, that means you don't get cortical rotation. That means there is no beta-catenin. That means it's ventralized. So in a ventralized embryo, there is no goosecoid. If you do the opposite, if you treat with lithium chloride inhibits GSK3, which means beta-catenin is everywhere. So if beta-catenin is everywhere, goosecoid is going to be everywhere, so you have lots of goosecoid in a dorsalized embryo. Okay, so if you inject it, you get double axis, it's found in the right place, it goes up and down as you would expect it with dorsalized and ventralized embryo. So it is an important molecule in the dorsal organizer. Cells around it become more dorsal. So we're good? However, goosecoid is a transcription factor. For it to dorsalize things, it can't signal. It's a transcription factor. You need other signaling molecules. So when you look at cells that are in the dorsal if you look at molecules in the dorsal organizer, there's lots of things present in the dorsal organizer. But what you need for it to signal, you need things that are secreted. Okay, so things like C1, twin, and goosecoid, they're nuclear proteins. They're not going to be involved in signaling. They are going to make products that are involved in signaling. So we are going to now look at molecules that are in the dorsal organizer that are involved in signaling. So things like noggin, cordon, polystatin, Cerberus, Frisbee, and Dickoff. So those are some treated molecules that are found at or near the dorsal organizer that play a role in making everything around them dorsal or neural. Do you remember two of the jobs of the dorsal organizers is to dorsalize mesoderm around it and to make ectoderm, neural ectoderm. So therefore, the dorsal mesoderm, that notochord has to secrete things. It secretes lots of things, and we'll look at some of them now. So far so good still? All right, so noggin. So just like with goosecoid, you can kind of look at noggin as the same way. Noggin is an important molecule. Noggin is a secreted molecule, but it's going to bind to receptors, and it's going to make things more dorsal. right? So it's found in the dorsal organizer, first of all. So if you look at sort of in situ hybridization, where is noggin? Noggin is found in the dorsal organizer. Then as gastrulation occurs, noggin is found in the notochord, right? So noggin is found in the notochord where you're expected to. The notochord is dorsal mesoderm. The notochord is secreting noggin. The reason it's called noggin is if you overexpress noggin, you can get a bigger head because you're making more sort of dorsal structures. Okay? It can make things more dorsal. It can change ectoderm into more neural. It does the things that you expect a protein, a secreted protein from a dorsal organizer to do. It can also rescue ventralized embryos. So if you remember I mentioned you can treat an embryo with UV light and you ventralize it, or you can treat it with lithium chloride and you dorsalize it. By using different doses, you can actually make a little gradient of things. So here's a normal embryo in the middle, a normal tadpole. If you treated it with high levels of ultraviolet light and you did not allow any cortical rotation, you end up with a ventralized embryo. And this is sort of a ventralized embryo up here, where it's basically just all yolk. There is no dorsal. If you treat it with lithium chloride, it tries to make everything dorsal, so it tries to make everything dorsal mesoderm. Okay, so here's normal, one side is ventralized, the other side is dorsalized, and using different doses, you can kind of go from ventralized to kind of a little bit of an axis to a normal axis, and you get more and more dorsal axis. But you can use molecules such as noggin or glucosid to rescue things. So if you take an embryo and you treat it with ultraviolet light, you will not have a dorsal organizer and you will get a ventralized embryo. However, if you take that embryo, treat it with ultraviolet light, so you got rid of a dorsal organizer, but then you inject glucosid or you inject noggin, you can rescue the axis. You can rescue, and you might get an embryo that looks like this, where it's trying to form a dorsal axis, it just can't. Okay, so you can rescue a ventralized embryo because it gives its dorsal character. Okay, so noggin, important molecule in the dorsal organizer, it's a secreted protein, as is cordon. Cordon, as the name suggests, is found in the notochord, which is the dorsal mesoderm. So it's found in the dorsal organizer, which becomes the notochord. So cordon, just like noggin, everything the same that's true about noggin is the same as cordon. It can make a second axis. It can rescue a ventralized embryo. If you dorsalize an embryo, you'll get more noggin. If you ventralize an embryo, you get less noggin. Right? So it does, cordon I should say, but they're almost the same. And indeed, they have overlapping functions. A lot of these molecules have overlapping functions because dorsal mesoderm is very important. So there's redundant mechanisms. So noggin, cordon, and to some extent follistatin are all doing the same thing. Redundantly making things more dorsal. One thing I forgot to mention that noggin does, it binds to and inhibits BMPs. So noggin inhibits BMPs, cordon inhibits BMPs as well, and another molecule is follistatin. Same idea, follistatin inhibits BMPs. Okay, so all of these molecules can make things more dorsal. These molecules are all secreted and they are involved in making embryos or the cells more dorsal. However, people noticed something unusual when they started playing with follistatin. Okay, so if you remember I mentioned before this sort of experiment where you can take the animal cap, so you can take this ectoderm, and if you take ectoderm and you leave it by itself, it basically stays as ectoderm. But if you take ectoderm and you treat it with different levels of activin, and the reason they used activin because that was sort of the first thing that they identified. So activin is in here, sort of right next to Nolan veg 1. It's kind of similar molecule. So activin is going to trigger sort of dorsal development, and if you have lots of activin, you can make dorsal things like the notochord. If you have less activin, it's more lateral things such as muscle, and if you have low levels of activin, it's more ventral things such as blood. So people knew that you can use these molecules, you can use activin or these TGF-beta molecules to do this. So if you take the animal cap, so this is ectoderm, you take an animal cap by itself, you get epidermis. If you take an animal cap and you add this TGF-beta family member, such as activin, you get mesoderm. However, if you take a TGF-beta family member and add follistatin, you end up getting neural. So something weird is going on, right? It's sort of strange that you're getting this neural formation where you're expecting to get the mesoderm. Having said that, one of the functions of dorsal mesoderm is to make ectoderm more neural. And this is how sort of people figured it out. Okay, so what's happening here? Okay, so ectoderm actually needs to be signaled to form epidermis, right? If epidermis is not signal, ectoderm forms neural. So the default for ectoderm is neural. So back to this little experiment